Yeah, cut my portion out.
1, 2, 3.
Uh-oh, here we go.
Whoa.
Wow.
Hello, everyone. Good to see you all joining us.
Hello.
Welcome.
He's muted.
Feel free to share your screen. Hi, Selena.
Hi.
Hey, Dina.
Hello, where's it going?
There we go.
Hello, everybody.
Your mic's off, Dina.
Hello.
Joining us.
Yeah.
Share your screen, share
Hello.
your audio.
Yes. No.
Welcome to share your audio. You're welcome to share your screen.
All right. Good
We
evening.
would
There we
like
go.
for you to visit with us like we're having a backyard barbecue.
Hey,
Those of you
Kim.
that are not quite so
There we go.
familiar with Zoom, you can go to a speaker view at the top right-hand side,
Goodness, what's wrong with me?
speaker view or gallery view, so that you can see one person instead of everybody. So please feel free to do whatever is good for you. More people are joining us, so we will be working on admitting everybody. If something happens to your service, your screen, your ability to see us or hear us, then I would suggest that you go out and come back in. We're not trying to restrict anybody from being able to listen or to see. So, please feel free to do whatever you need to do to join us. As we begin and people start talking, then you might mute yourself. That way there's not the noise that's coming on.
There. Can you hear me now? This is Susan.
Yep, we can hear you, Susan.
Okay. You can't see me, I think.
No, I can't
No.
see you, but you're welcome to share your screen.
How do I do that?
Well, there on your bottom left-hand side of your black part of your screen,
Yeah.
the far left says mute, or it says start video. So click on
No.
your start video,
There
and
we
that
go.
should let you pop right in for us.
Okay.
So-
That's not working. I mean, there's a round head and a top. Yes, but I can't make it do anything else.
A little bit more. There.
I would suggest keep trying.
Whoops.
Maybe that will pop up. Can you see all of us?
Oh,
I can see two people. Now I
Okay.
can see a rug and somebody walking across the rug. So up at the only thing I have is Christy Hewitt left, and I have in the upper right hand corner I've got record, I think, or REC, which I assume is record. And I can see your two faces, and then I can see this Susan Parkinson in the round circle and a plus and a minus. That's all I have.
Okay, so are you on a mobile device or are you on a desktop?
Oh, yeah, I'm a mobile device.
Okay, well there's a button on there that lets you go gallery view or speaker view, and most mobile devices are so small that you pretty much only get one or two people in your screen.
But in terms of economic theory.
So, please feel free to just kind of dance around there and see if you can find the
Okay,
right button
there's
that would let the camera.
share screen.
They ask all the students, what would you?
Somebody has got their news on, and we can hear that pretty clearly. I would ask that whoever has that on to maybe please try to mute that, or mute yourself, please. It is so wonderful to see all of you tonight. Thank you all for joining us. We are so proud to see all of you. This is absolutely fabulous. So
Yes,
I'm
it
just
is. Whoops.
I'm just going down through here and asking people to unmute and to share your screen. So please feel free to do that. We would really love to be able to see all of you. If you can't, we certainly understand that you can't. But you might be able to try. If not, then maybe we can schedule another time where we can get with you and maybe show you how to make that work.
Okay, I'll do that. I'll just talk, but I can't see it. I mean, I can't. You guys can't see me, but I can see you. So
Okay, good.
I'll be
Well,
quiet
that's good.
now.
Where am I?
That's
Bonsoir.
good. There's so many people on our screen tonight that, we can't see everybody with one glance. And so there's at least two full large computer screens that we have visiting with us tonight. So thank you all for joining us. I'm Norma Pruitt. I work with the foundation and helping to do whatever needs to be done. And we've got a pretty good little robust, informal kind of an agenda for you guys. I see Dr. Fink trying to load on here with us. Corey Brosted is with us. Several board members are with us and some committee people that work on our committee. So thank you all again for joining us. I would like to just kind of remind you some housekeeping rules more than anything. If you've got noise going on, please feel free to unmute yourself. To mute yourself. If there's a point where you've got a question, then jump in, give us time to hear the question. There's like 50, 60 people on our screens. So thank you again for joining us. What we're here for is to talk to you guys, just to basically say hello and visit with you and talk with you like we're kind of sitting all together at a backyard barbecue. Now, we're located in Kentucky, and it is really, really cold, and we even had snow flurries today, so we'll have an indoor barbecue, and we certainly would want to invite all of you to participate. So you have a speaker view and you have a gallery view, and feel free to pop that and toggle that however it works best for you so that you can see who you might want to see talking. And again, just jump in when there's a particular question. So what I'd like to do is to remind you that what we're here for is for Giving Tuesday. And for those of you that are not quite familiar with what is Giving Tuesday, you may be just new to the SPF Foundation, and that's fine, that's wonderful. And some of you have maybe been with the foundation and know the work of the foundation for many years now. We want to help you to understand tonight what is Giving Tuesday, because that's tomorrow. We want to talk with you about Giving Tuesday, and then basically some casual conversations about what is the foundation and what do we do with the foundation and what do we do with the money with the foundation. So I've got my better half with me sitting right here, which is Greg Pruitt, and he's going to talk to you about what is Giving Tuesday. And then we can come back in and just kind of visit here around the rooms and talk after he's finished.
Well, thanks, Norman. I'm just going to take a couple of minutes. Obviously the words Giving Tuesday kind of give that away. But when you think about this weekend, when I was a kid, Thursday was Thanksgiving, Friday was Black Friday, and that was an extra day off and football in the yard. And then it turned into the day I had to put up all the Christmas decorations and take care of kids while we did that, while somebody else shopped. Then Small Business Saturday came along, and we, get out to the communities in our part of the world, because we live in a world of small communities, and try to do a little shopping and supporting. And then Cyber Monday. And for those of you who know me well, you know that Cyber Monday, the best place for me to do is get over in my corner and let Norma handle Cyber Monday. So that's what I do. But then Giving Tuesday comes along, and that's what we're here to talk about just a little tonight. What is Giving Tuesday? Well, it's a global generosity movement that unleashes the power of people and organizations to transform their communities and their world. And as we think about what that is, what is our community? Well, this community is a community of individuals and families, caregivers who all share in the world of dealing with hereditary spastic paraplegia or primary lateral sclerosis. And so as we think about Giving Tuesday, we want to think tonight in how we can improve through Giving Tuesday this community that we're part of. Giving Tuesday was created in 2012. And it's just a simple idea that encourages people to do good. As we think about this foundation and our communities, I would just ask us all to think about doing good in a couple of different ways. And by the way, Giving Tuesday is always the first Tuesday after Thanksgiving. And of course, this year falls on December 1, but each year it will be the Tuesday after Thanksgiving. But what are a couple of ways that we can give and do well for our community? Well, when we talk about giving, we always think about money, and I'll hit on that in just a moment. But just as important for this group is we need you to be involved in helping us improve and increase awareness of our conditions and our diseases and the opportunities that we need to enjoy to get to know one another. As I look at all the names and faces on these screens, a number of you I know and see from time to time on social media. And it's always good to talk to you, see where you are and what you're doing. And so that creates strength within our foundation and our organization. If you're out there and you see people in this group that you don't know, but you're on social media, look for us. On social media, I'm Gregory Don, and my symbol is HSP Kentucky. So if you find me, ask to be a friend, let's get to know one another. And let's strengthen this organization through giving of ourselves in time and discussion and support. And what's the other way? Obviously, we always are interested in trying to raise dollars. Why? For one purpose, medical research. You know, the vision and the mission of this organization is to raise dollars to help find treatments and cures for these diseases. As I look at the screens, I see people of many different stages in life and circumstances. I'm sure we're all in different stages of dealing with the disease. I'm here primarily because what I have, my children and grandchildren have a 50% chance of having to deal with. And I want to help them deal with this better as they watch what we do and how we raise money for research in the future. And we've got some folks that'll talk in a few minutes about that research and about raising that money. There are ways you can do that. Go to our website. You can find ways to plug in with your time and with your dollars. So Giving Tuesday, again, we appreciate your being here with us tonight to talk about it, and let's get to know one another better as we work together to make a difference. All right.
Hey, that sounds great. Thank you, Greg, for talking to us about that. And because you guys know that you can look on, just Google Giving Tuesday, and you can find all kinds of statistics about Giving Tuesday and how much money has been raised just globally. And of course, you know what is near and dear to our heart is this foundation. And that is what we want you guys to understand, is that we're all working together for this foundation, so that is the direct beneficiary of the work that we're doing. And as you guys know, we have looked at annual conferences, and we have done the in-person conferences, and our attendance has grown, tremendously grown. And so this year, as you guys know, we couldn't very well do that. And we had the virtual conference, and we had a lot of people participate in the virtual conference, listening to what the doctors, had to tell us about new research and things that's going on in the medical world. And we have those videos on our website. So you can feel free to dance around all over the website. And here later, I will show you some sites about the donation buttons and things that we can do to raise money individually for the foundation. And I will share that information a little bit later on in the program. But what I'd like to do right now is I'd like to introduce to you Frank Davis. Frank is the president of the foundation, and he works very hard in helping to keep lots of subject and lots of balls going. There's lots of information that's out there, and he helps us to discern what is really valuable for us as a foundation and as individually as we're dealing with all of these diseases. So I don't see Frank on my screen because we've got 3 screens of people that have joined us tonight. Again, I want to thank you all for joining us. So Frank, I see you now in the top corner of my second screen. So thank you so much for joining, and take us away.
Okay. Thanks everybody. I've been combing through the faces on my screen, and for some of you, it's good to see you again, and others, it's good to finally put a face with a name. So welcome everyone. I wanted to, I don't really have a speech prepared, but I wanted to go over a few things and then just be here to answer questions if anybody wants to ask them, because at a barbecue, it's very seldom that you have a speech maker. But I wanted to go over a couple of things, and a lot of it is when before I had this condition and before I knew I had this condition, I would give to charities from about once or twice a year, and there would be certain criteria I would have for whether I was trustworthy of giving to that charity. And one of them is how do they spend my money? What do they spend my money on? And how much waste is there? And that's what I kind of want to go over. And for a donations we're receiving, we have an anonymous donor that's going to be doubling every donation up to $200,000. And then how else is it improved? The other parts of what make us who we are is we're a 17-year-old, well-respected a patient advocacy group, and we carefully scrutinize every penny we spend because all of us are volunteers, and to decide what research we support, we have a scientific advisory board that carefully scrutinizes every research proposal we get, and then 2 or 3 of the people on the board will analyze each scientific proposal, and then those grades are put together by the manager of our scientific advisory board, and then she puts the grades together and reports to us. And we have not supported anything that's less than excellent and very promising for years and years. And if I were a donor, I would like to know that and know that my dollars are being well spent. Additionally, when we pay to support someone, it goes on their record. And with our donation being on their record, the National Institute of Health is much more likely to fund them again. And they often, after getting a funding from us, will be supported by the National Institute of Health in many times more than we've supported. For instance, Dr. Zuckner once used our funding to discover a couple of genes with HSP. And then because we had supported him, he got an additional several $1 million from the National Institute of Health. And that helped him discover many more genes. And so whoever donated that year, their money was multiplied hundreds of times. And that happens every year. If I we send out an annual report every summer, and last summer it had 8 pages of just the studies that we're sponsoring now. And I realize that is very difficult to understand, and it's even very difficult for lots of us to understand. But I ask each of our scientists to turn their report into me in the language of a 13-year-old. And even then, it's too hard for most people to understand. And but even scientists will admit that it can't be translated any simpler, or it would have to be 50 pages long because of an explanation for every word. So anyway, last year, there was a study published about HSP or PLS twice a week, every 3 days of last year and this year as well. So you know that the science is moving fast, and all of that is exciting, but it demands to be sponsored and paid for. We don't, as I said, don't support anything that's less than excellent and very promising. And for many years, we've had many more proposals that were excellent and very promising than we could afford to support. And so if anybody's wondering about whether to add to what we're doing, please know that by our funding growing, our potentiality will grow astronomically. So it's really important that we can do more if we have more. So, and again, it's all going to be doubled. So if anybody has any questions, let me know. Thanks.
So as you guys can hear and see from our president, Mr. Davis, there's a lot of work that's going on. And as you guys all know, we are all volunteers trying to pull our train. So we really need for us to work together and see what we can do to raise our voices so that we can be heard and be seen. And so that's one of the things that we're wanting to do with our new Zoom platform is to give there's the opportunity and the time that we can all visit and try to learn from each other, the things that benefit you, the things that can benefit the foundation, and how can we be a better foundation. As you guys know, there's a chat screen. For those of you that are on a computer, you might be able to see this chat screen that's kind of scrolling along over on the right-hand side. If you haven't seen that yet, then down at the very bottom is the word chat, and you can click on chat, and then that chat box will pop up over here on the right-hand side. So feel free to reach out and ask questions out there if you would like to, and then we can call on you and let you have a chance to do that. So I think Terry has asked a question.
Yeah, so first of all, most people know me on the forum or on the group, and I want to say hi. I haven't met you in person, so we'll meet virtually. I just want to thank the foundation because without your support, we wouldn't be where we're at right now. And if you know our story, we're working on a gene therapy for SPG 50. And many years ago, this amazing foundation supported research that led to us to get to the point we're at. And if it wasn't for this amazing foundation, we would be nowhere near the progress we're at right now. So I just want to first off thank everybody for supporting us and contributing to, I know that SPG 4 is a massive SPG, and supporting our small little one at the time meant a lot, and it allowed us to leapfrog into the gene therapy platform that we're working on right now. So I just wanted to thank everybody.
Thank you, Terry. We appreciate your words. We appreciate the work that you're doing and the commitment that you guys have put into taking care of your little one. So thank you very much too. I think we would all like to just kind of give you an applaud.
And on top of that, I really want to say that, we can do some amazing things. Anybody that knows me knows I'm helping, I'm lost track now of how many families I've helped out, but I truly feel that we can eradicate these diseases. And unfortunately, the reality is it's all about money. And if we can come up with enough money, we can eliminate these diseases. And I'm proof of that. We're already into our gene therapy. Next year we're going to be applying to the FDA. So, we can do this for SPG4. We can do this for all these larger diseases where, that you are afflicted by. And I don't know how we do that. I don't know, how we garner more support so we become like Rett syndrome or Duchenne's muscular dystrophy or something where people really take notice. But I think we need to do that because we have an opportunity with gene therapy to change our lives and the lives of our children, right? So I just want to say that, I'm here to help anybody at any time work through this. Dr. Fink has been amazing to all of us, to giving us his time and supporting our families. But, I really want to help everybody. I want us to change the world and make it so that, everybody on this forum does not have to suffer anymore.
May I ask?
Not be truer words.
May I ask a question?
Sure, Susan, go ahead, please.
Okay. As far as this donation goes, does that go through to January 15th, or does it have to be donated before 2021?
Frank, do you want to answer that at this point? We're going to get into the donations, but do you want to specifically answer her question about the donation for, I guess, the match?
Yes.
Okay.
Okay. In answer to your question, the donor is matching donations, any donations that's received between November the 15th and January the 15th. And,
okay, great.
So if we donate tomorrow morning at 8 o'clock, 8:01, and would that mean that we get triple the donations towards the foundation?
it's just being doubled as well. So, but that'll be counted toward Giving Tuesday. And so Giving Tuesday is kind of an exciting day, and people want to participate on that day. And so we're shooting for a goal of reaching $88,000 tomorrow. So,
and the reason why I'm asking, Frank, is that if you donate, I'm not sure if you know, if you donate on Facebook, and everybody does it at 8:01. Facebook, so you're going to donate 100. Facebook's going to donate another 100 to make it 200. It'll get to the foundation, and then they'll double that, making it 400 potentially, right?
We've tried to work with Facebook in the past, and I don't want to discourage anybody, but what they finished doing is gone in a flash of a pan. So I don't know that we've
gone in like minutes, like 2 minutes.
Right.
It's gone.
Right.
Exactly.
Right. So I don't.
But I'll be there at 8 o'clock in the dot, clicking submit, so that we get the quadruple, right? So.
Okay. Good luck.
And one of the things that we're going to talk about here shortly, Jim Sheehan is our fundraising chairman, and he works with the donations, and he works with different things to try to find grant dollars and try to find additional resources to help with things. So if you guys have any ideas that you could, bring to the foundation that we could all collectively work on and work together with, Jim, he started a 5K back a couple of months ago now. Time is flying. And he raised up, quite a bit of money doing this 5K. So he's going to talk to you about donations and how donations work with the foundation. And then we'll talk about some more things as we go along. So Jim, I see you up in my top corner. Can you take it away?
Great. Thank you so much. Hopefully everyone can hear me. I just wanted to kind of give you some things that are going on with fundraising. And if you guys have any questions about fundraising, have any ideas, have any criticisms, feel free to send me an email at fundraising4spf@gmail.com. That's a separate email that I've got set up for this. But we've talked a little bit about Giving Tuesday. We talked about the year-end campaign. Again, we know that whether you give tomorrow or anytime through the 15th of January, your donation will be matched. So that's awesome. And I think Norma's going to go through and show you a little bit later how we can set up different fundraising pages for you, like we did as she mentioned, the virtual 5K back in August. And we had, I think, about 46 people that participated and raised money. We raised about $25,000. That was a huge success. We weren't thinking it would be that big. So it was just great that those that wanted to get out and get some exercise could raise some money, get some exercise, take pictures. We had little race bibs that were printed up that everyone got to wear and take pictures and stuff. So it was really an exciting time to get out in this crazy world we live in and be safe, of course. So hopefully we'll do a bigger and better one next year. So look forward to information probably late spring, but it'll be the third week of August. That's the week of our awareness week. So it'll be that week. So hopefully you can do it sometime in the month of August. And you don't have to do a 5K. 5K, for those who don't know, is 3.1 miles. If you work out with a therapist for an hour, call that your 5K. If you walk out to the mailbox 10 times in a week or whatever, just kind of calculate what it takes you to do some kind of physical activity for about an hour, maybe an hour and a half, and then call that your 5K. It's not that big of a deal. It's on your honor anyway. And then reach out to family and friends and raise money. So another event that just happened, and it's actually still going on now. There's a little girl named Blair. Her name's Blair Bear, and she's two and a half years old. She has HSP, and her mom and her dad got together and wanted to do a virtual 5K for her. And their initial goal, I think, was $5,000. And at first it was ramping up really high. I said, why don't you raise your goals to say $10,000? She goes, okay. Ramped up higher. She went up higher. Long story short, they have raised over $35,000 as we speak. And I think they have until December 15th. So again, Norma, I think is going to show you how we can set up a page. If you have an idea, if you want to do some kind of fundraising event, we can set up a page for you. It could be your event. It could be for your family and friends to participate. Again, raise awareness and raise money. A couple of things I want to share with you. I'm not sure if you are aware that we have a monthly giving link on our webpage. You may wonder what that is. Well, you can either give a contribution monthly or quarterly. You decide what it be, $5, $10 a month, whatever, or quarterly. Go to the link on the webpage and set that up. then it's automatically done for you. Some folks like to have things laid out and paid on a monthly basis versus waiting to the end of the year. Some people may find it hard to save up money at the end of the year, especially if it's a rough year like this year or Christmas time, stuff like that. So if you want to do something like that, again, you can set it up monthly or quarterly. And your choice, you can cancel anytime. And you'll still get a letter at the end of the year with your final contribution. So that'll help out with taxes. Another item is planned giving. If you have a will or are planning on putting a will together, I hope you will consider putting the Spastic Paraplegia Foundation in that will. You can check with your financial advisor on all the details on how to do that. And then if you'll go to the Give Now link on our webpage, there's some information there about that as well. So if you've already contributed this year, great, thank you so much. If you have not, we'll hope that you will make a contribution, do your best so that we can fund some medical breakthroughs for us all. And we look forward to sharing some success with you about that. That's all I have. If anybody has any questions, feel free to let me know. And if something comes up later, again, fundraising4spf@gmail.com.
Thank you, Jim. I have already added that email into our chat box, so feel free to scroll down through those chats. And if you've got a question and we don't see it, then just ask it again, because that may get to be a little faster in the scroll. But what I would like to do is, Chris has told us that Blair is on the screen somewhere in one of these boxes. I would like to reach out to you, please, and get little Blair so that we can see her, please. And I think she said that they are up to $40,000 on their fundraiser. So, I mean, is that not awesome? I mean, let's just go, big clap. Yay! Thank you, thank you, thank you.
Hey guys, how you doing? This is Blair here. So, she's been raising a lot of money out there, and we're happy to be able to support the foundation. We just found out about the diagnosis this year, so it's been exciting to get involved. Sorry, we're in the middle of dinner time here with the little kids, but yeah, it's been really exciting. We've gotten a lot of donations from her school and family and friends, and her mom has done an excellent job of setting up the fundraiser. And I know working with a lot of people on this call, including Hank and Frank and a number of people. So thanks so much. It's been a great community that we've been introduced to here, and Dr. Fink and everybody that's supporting us and the foundation. So we're very happy to be able to participate. We hope we can continue to raise some money and make advances for everyone with HSP and PLS.
And just as a side note, if anybody is interested in, doing a kind of similar fundraiser to what we do, if you think you've got a big network, we basically leveraged our network and had them leverage their networks. And I mean, it helped that it was a new diagnosis, I think. But I'm happy to talk to anybody about our approach if anyone's interested in trying to put on a similar kind of 5K race or any kind of fundraiser, really.
Thank you.
Thank you, thank you, thank you. You guys are an inspiration, and Blair, you are adorable, and thank you so much for working with us and helping us, and enjoy your dinner.
Thank you, Blair.
Thank you for
Thanks, guys.
jumping on so we could see.
Thank you.
Let me say one thing more quickly about fundraising that they kind of hit on there for a moment. Fundraising can be anything from, if you can afford $5 a month, that's a monthly gift, that's $60 a year, and a bunch of those add up. That's wonderful. There are a number of you that may be involved in different activities. Jim did the virtual 5K. There are a couple of us, I know Board Member John Cobb, who I think is on here with us. He in the past, and then Norman and I as well, we've done golf events. My point is, if there's something that you like to do or used to like to do, and you've got friends who do it with you, it may be playing cards. It may be Tim and Tina did a trivia night for a while. If you have an activity like that you'd like to brainstorm and think about how to make into a fundraiser that you might enjoy, and it might could raise some dollars. If you need people just to brainstorm with you about that, contact Norma and me, or contact Jim. We'll think with you and try to help any way we can from a distance, help you put that together. And once you do it the first time, the second time, and the third time is easier. And what we've seen with those kind of events is each year they grow in terms of how many dollars they raise, because more people find out about it and get involved. So if you have an event like that we can help you with, please let us know.
that was such an inspiration to see little Blair and the family, and you know, they're coming together, you know, having just been diagnosed, the little person that she is, I think, what, 2.5 years old, you know, and they're in a whole new lifestyle and battle that we all deal with every day, right? So again, thank you all for joining us. Is there anybody that has a particular question of the things that has been said at this point? I was going to get into the website and the Classy platform that we use for fundraising, and to talk with you a little bit more about what Jim and what Greg has said. But I'm willing to open up to let anybody say anything they want to say. Okay, great. You're saving time for me. So here's what I'm going to do. For those of you that can see the screen, Hank is our technical guru up in the top corner of my screen, and he's going to do a share screen, and he's going to show you guys our website. So we've been talking about what Blair put together. She contacted our board members, and there's a number of board members that are on the platform with us tonight, and we'll get around to introducing them, and you're going to hear some more discussions from them. But what they did is they contacted us and said, we really want to do this fundraiser. We need help. What do we do? So what we did is when I say we, I mean we, the foundation, we have our own website, of course. And on that website, we were able to do all kinds of things to help talk about our donations and our fundraising efforts. One of the best and easiest ways to donate to us is to strictly go to the website. But what we did is we have a Classy platform that will let us help you create something as what you're seeing right now on the screen. So the family got together, gave us a few photos, gave us a little bit of some language, and then we create this platform on the Classy platform that allows you then to go to your family, friends, your work, whoever is your support group. And as you can see, these people have gone in there and they have loaded up their identity, if you will, created a team in some cases, and just some are just family and friends. So you can see the money that they're raising. So then when you get to the whole entire campaign and a little bit of $1 here and $5 here and $100 there, it has raised $40,000 in no time at all. So we as the foundation, the people that are here behind the scenes working, we can help you set this exact same kind of thing up. So we create a name, we create a goal, and then you just kind of blast out that link, as you see at the very top up there in the browser, and then people just start donating to it. So, you know, obviously little bitty kids always seem to capture your heart first. But we still want to help folks understand how to do these kinds of fundraisers. One of the things that we've always done is we've put together a golf tournament, and we leverage some of our sponsors to help donate, to help pay for people to come play golf. Well, this year, you know, all that's really kind of changed, but we did do an outreach, and there are people that have actually donated because we did an outreach, and they are willing to do that. So now we're showing you our website. And what I would like to do, just go back to the first white page there, Hank, and show them the red button. As you see underneath there, SPF Vision, there is the donate button. And every platform is a little different. It can't get much bigger than it is. But anyway, if you hit that donate button, then it will take you out to another one of our platforms that's like a network for good. And that will give you an opportunity to set up either a one-time donation, or you can put it on a monthly. So you could, you know, donate $5 or $10 or $150 or whatever it is that your budget will allow you to do. You can go in there and put that in there, and you can choose whatever you would like to choose. So that really is one of the best instant ways for us to get money without having to go through another third party. Now, some third parties charge a % of everybody's donation. And so, you know, I'm not just trying to say we're trying to be stingy, but we really want to collect every penny that we can collect and not give it to another third party, because we need that money for medical research. So I wanted to share this with you so that you can see that. That way you've got an opportunity. If you don't really want to set up a time for the Facebook and let Facebook go through its process, and then Facebook eventually, you know, sends the money to us, we have a way that we can help you set up the Classy platform, which is a little bit more instant, or you can just do a direct donation on site. So as you see, when you look at the donation tab, donate here, there is also the very old-fashioned way of just mailing in a check. And we have the address under that tab so that you could just pop a check right in the mail, and it goes to our home office, and that way it just is basically deposited instantly, and there's no issue, and there's nobody digging in our pocket for any kind of extra percentage of funds for handling it. So I wanted to share that with you. I wanted to give you the opportunity to see these various different kinds of ways that you can donate by credit card, you can donate by PayPal, you can do eBay. We also, if you know people that work for the federal government, it is so easy through the CFC program that they can actually just have that money direct deposited right out of their check, right into the fund, the CFC fund, and they can then send that money to us as well. And then also, I don't think we have on here that I can see at a glance, but for those of you that are very savvy and wanting to buy your Christmas gifts on Amazon, there is the platform that's called Smile, and it's smile.amazon.org. And you can go there and allocate that you want any extra money that Amazon will donate, that it will come to this foundation. So I think somebody has already popped up a link for us there. So please feel free to look at your chat boxes, and that way you can be led to that actual place on the Amazon platform. So anyway, there's lots of information that we would love to share with you if we had time to go over all of the website, lots of useful information. And if you have any questions about any of that, you can reach out to any of us, and we'll be glad to walk you through, show you exactly the link that you're looking for, and help you to find information. Because I really believe, strongly believe, that the more awareness that we can raise, naturally we're going to raise more funds. So if you can help us in raising awareness, and what we like to use is that hashtag HSPANDPLS, because we are a foundation raising money for both motor neuron diseases, then that helps to spread the word on social media using hashtags. So if you just go and do hashtag HSPANDPLS, then you're going to be able to see all of the social media platforms that we're currently on. So please feel free to reach out in any of those ways, and to you know, share the information that we're trying to share so that we can raise more awareness. So I've thrown a lot of information at you. I really wanted to kind of give you just a thumbnail of the platforms and the ideas that you might want to come up with. You might do some kind of a creative Christmas kind of thing. You know, we can add some Christmas decorations and some Christmas graphics. So please feel free to let us know if you guys have an idea for raising money. Jim is our fundraising committee chair, and his email address is over in the chat box, and we'll be glad to get back in touch with you and talk with you about what you can do to do a fundraising. Are there any questions that particularly jump out at somebody as we're talking about this? Don't be shy.
Norma.
Yes.
Norma,
Yes, Frank.
I'm not appearing, but anyway, I noticed that Dr. John K. Fink is in attendance, and wanted to perhaps introduce him so that at least everybody could, if you haven't ever seen him or heard his voice, he's here tonight. John, are you here?
Frank. Thank you, Norma, and thanks everybody. It's really just an honor to be here. It's very exciting. The whole field of spastic paraplegia, that is primary lateral sclerosis and hereditary spastic paraplegia, really didn't exist in this way as 30 years ago. 30 years ago, there were a couple of conditions grouped together, but no genes really had been discovered. The concept of gene therapy 30 years ago was just a fantasy. And now it's moving into many laboratories around the world and in the United States are developing therapies, both gene therapies and chemical therapies. And the whole thing has been transformed, but it's not 30 years anymore. It's growing logarithmically. It's doubling now every year. So there's tremendous research advances this year compared to last year. And this is, it doesn't just fall on your doorstep. It's created one step at a time, one research project, one family study, one grant application. Now, the research is very expensive. And what we do with these kinds of fundraising is we bring in more investigators into the field. And besides new investigators, if somebody has an idea they want to test, well, NIH doesn't fund ideas to test. NIH funds projects that are very far along in their development and then will support them for the next phase of their development. For example, if you want to create a mouse model of a certain type of hereditary spastic paraplegia, you don't apply to NIH because NIH will say, after you've created the model and you've shown that it has these symptoms and you've tried some therapies, now you want to take that to the next level. So how do you get your project to the first level? It's by this kind of funding. This is the way that projects are bootstrapped, that they can sort of launch into that next level, as Frank was pointing out. So this kind of funding is absolutely essential for both new projects and new investigators. And that is what has been responsible for this logarithmic growth in the past several years. Now, there's too much to talk about and not enough time. And I myself, as you know, but I'll just mention, I host it's actually a support group that's become so large, it's now question and answers, but we're going to still try to get back to a support group. We may now have, but anyways, that's tomorrow at 6:30. And so it's the first Tuesday of every month, but not in the summer. First Tuesday of every month at 6:30. And I can answer as many questions as I can between 6:30 and 8. Or people can talk to each other and share contacts and relationships develop offline. And I mean, outside of the Zoom conference. And so that's tomorrow at 6:30 to 8. And I'm happy to answer any questions then if anybody has any specific questions. But thank you very much for inviting me.
Dr. Fink, while you still have the floor and we have your attention, thank you so much, sir, for joining us. Can you tell us how that we can actually get to your support platform?
Very good question. I think at this late hour, because it's tomorrow, so somebody could send me an email, or let's see, I guess that would be the way to go, would be to send me an email, and I'll try to pass that on up until showtime at 6:30.
Okay.
So if you send me an email with the subject matter, a subject line saying, please connect me to the support group meeting, something like that. I'll pay real close attention to screen for those in the next 24 hours, and I'll invite everybody I can.
Okay, Dr. Fink, thank you. So that you don't receive 70 emails tonight, why don't, Frank, if you don't mind, why don't you send that email to Dr. Fink And then we can, then we can get that information out to those folks that have joined us tonight.
The link is on our webpage.
There you go. Already at work. So if you'll go to our website, you will already see that link exposed in the events, the upcoming events. So you'll be able to find that information on the sp-foundation.org. Very good.
Let me ask a question. How am I going to get those? Are they going to be emailed to me?
The questions that everybody's asking?
No. When people want to sign up for the meeting, how am I going to get their emails so that I can invite them to the Zoom conference?
Well, what Hank just told me is that is on our website. So everyone should be able to go on our website, and Hank's going to pop that up right quick so that everybody can see this, where it is, where it's located. So they will automatically be registering with their own email address, Dr. Fink.
Okay.
So
I trust you. I trust you completely.
Thank you. I appreciate that. So whoever is managing your Zoom platform and they are asked to register, then all of these people that might be on our call tonight, they're going to go to this link as they can see it on our website right there, which is Giving Tuesday, 6:30. Everybody see it, go down one, Neurological Disorder Support Group. So if you guys will go to our website, link to that, there you go. You will be able to scroll down and find that link and be able to log on to that. So then you should be able to receive everybody's email, Dr. Fink.
Okay. Well, I'll tell you what, just as a safety check, why don't you and I talk tomorrow about 4 o'clock? Is that okay? I'll send you an email and just make sure that I'm all good with this. I don't want to leave anybody out. So I'll make sure I'm available. But I'll just double check at 4 o'clock. But anyways, thank you very much.
Okay.
Could I just ask a question? Is that Eastern time?
Central.
Central time.
No, 6:30 p.m. 6:30 p.m. Eastern time.
6:30 p.m. Central time.
Meaning Michigan time.
Eastern.
It's Eastern Standard Time.
Eastern time.
AKA Michigan time.
Oh, okay. Eastern time.
Eastern time, 6:30 p.m. Eastern time. Good question.
Thank you. And you guys can see it over in the chat box. Brianna has already added that into the chat box, so you guys can all grab that. Of course, we would really like for you to go to our website because that gives us a little extra traffic and we can then show that the statistics are reaching folks. So please dance around the website as much as possible. Thank you, Dr. Fink. We so much love you and appreciate you. One of the questions that I really wanted to ask you while I still have your attention, Frank had mentioned earlier about the number of SPGs that has been discovered. Can you give us an update on those at this point?
Well, I don't know. There are more than 80, probably 90 or so. Between 80 and 90, and that number is growing. I know of several that haven't been published yet. That's what I know of. So probably other investigators know of others too. So more than 80, probably less than 100 known types, but it's in that range. One comment though, and this is very, it's like family members. The people in your own family look like you to a certain degree, and your cousins sort of have a similar resemblance. By the time you get out to distant relatives, they may not look at all like you. They may have very little similarities, even though they are members of the same family. And that's the same way it is with the hereditary spastic paraplegias. Some look very much alike, and then others that are still considered a form of hereditary spastic paraplegia don't resemble the others very much at all. So the number will grow even beyond this, and it will grow mainly at this point, I predict, mainly with the extremes that don't look like, they're still forms of hereditary spastic paraplegia, but they're quite variant in their presentation. So So some of the types of hereditary spastic paraplegia, if you read about them, you say, well, I don't look like that at all. And that's true, because some of them are quite dissimilar. But at any rate, that's about the shortest answer I can give.
Thank you, sir. I appreciate that. I think one of the things that is essential and is highly important is that if you have not yet joined the SPF Foundation, which is, basically a free membership, it helps us to gather the information so that if in the future there is the request for those folks that have a certain SPG, then we're able to go into the database and actually pull that information out. So I'd like to encourage you, if you have not done that, to join the foundation in that information search as we all work together with this. So thank you, Dr. Fink. One of the things that I'd like to do now, if everybody is willing, is we have Mark Weber. He works with our scientists that are looking for funds to help medical research. And Dr. Corey Brostad is also on with us tonight. But I'd like to start with Mark. And Mark, if you don't mind, talk to us about, once we collect money as a foundation, what happens to that money? Where does it go, and what do you do with it, and who is it that you give it to help us? Can you share that information, please?
Sure. We fund scientists around the world. What we do, we put out a request for proposals. I have a email list of, right now it's right around 440 different scientists in North America and Europe. We put out the request for proposals, we get proposals. We then send the proposals to our scientific advisory board. Right now, we have a batch of 20 proposals with our scientific advisory board, and in about 15 days, we should be getting the results. They're tabulated from best on down. We fund the top rated proposals until we run out of money. And then I have the truly awful job of telling researchers who didn't get funded that, sorry, you had a great proposal, but we don't have the money. So how have we done funding proposals? We started funding proposals in 2004. We have funded $8.4 million of research. Last year, 96% of every penny we got went to research. Under 1% went to fundraising. The rest went to administration and educational programs. We have funded so far 66 projects in North America and Europe. We funded researchers in major institutions: Harvard, University of Michigan, Stanford, Berkeley, Sheffield, Oxford. We've funded proposals all over the place. Trying to think of what else I can tell you. That's pretty much it in a nutshell. Does anybody have any questions about our research, what we do?
I have one.
Yep.
May I just ask you to confirm again, what percentage of donations go towards these projects?
90. Well, I just looked before, like about 10 minutes ago, at our IRS filing, 96%.
96%. That's fabulous.
Yeah.
That just shows all of you working to make this happen. I appreciate it.
Yeah. It's a lot of volunteer hours by a lot of really talented people.
I have a question. I look on clinicaltrials.gov quite often to see if there's anything. My husband has SPG 7, and I see so much that's of research that's going on, like just right now in the Netherlands, in China, in Italy, in Austria. I don't see that much in the United States. Is that because this is just the Are the ones that are out of the country, are those funded by NIH?
No, not to my knowledge. You're talking about actual clinical trials though, right? Drugs.
I'm on clinicaltrials.gov.
Yeah,
Yes.
so that would be drugs.
Of the reports that you're looking at, how many are actually clinical trials and how many are just studies in evaluating what the diseases are? Because a lot of times, even though it shows in clinicaltrials.gov, they're not actual studies in the actual drug. It's more like natural history study,
Right.
right? So you have to really look to see which ones are actually a drug one. I know there's a few out of Australia. There probably is a few out of Europe.
Yeah.
Well, where do you look up ones that are for a specific type of SPG?
You
Like
go
say,
to the website, you punch in the gene or the disease, and then you look at the ones that say enrolling, and then specifically translational science of some sort or delivery method.
Okay. Thank you.
Yeah, we are, there's a group of scientists down in Australia. They have a drug ready to roll. The problem is, before you do a clinical trial, you need some sort of objective way of determining whether it works. And they are in the process of developing biomarkers. Once that's developed, then they can go forward with the drug test. But before, it's just a waste of time and money.
As you can imagine, if you repurpose a drug, so say an existing drug exists and you have to repurpose it, you still have to show that it's efficacious. That's actually going to help the person doing the disease. If you're repurposing a drug and then you have to make it, say, for example, say you find a drug that affects your liver and you need to make it so that it affects your brain. Well, they're going to have to recompound that drug. So in order for it to not affect your liver or give you some other disease or maybe affect your kidneys, make it so it's more susceptible to affect your brain or the person's brain that's being affected. If you're doing a gene therapy or a completely new disease, you have to then show it in mice. You then have to manufacture the drug. You then have to test it in rats or potentially monkeys. And then you have to apply to the FDA. It's a long process. It's not for the faint of heart. It's just, and the reason why is when you look at diseases, no drug company will touch a drug that's less than 1,000 patients worldwide. They won't even go near it. And even then, that's assuming that a patient organization has done at least a little bit of the work, right? The drug companies are looking for drugs that are over 200,000 patients or diseases that will make them money, right? But if you kickstart it though, if we kickstart it, then some companies will get interested. That's the way it works.
And there is rare disease designation for drugs, and there are certainly a good number of drugs that have been approved with fairly small patient bases.
Yeah, and the reason why just so everybody kind of understands the economics around it, if you do a gene therapy, the company's going to want to charge $3 million a person because insurance will pay that today. The problem happens when you're in like Canada or India or Pakistan or somewhere else in the world, and the government won't pay for the drug, and then you have to fund your own $3 million worth of funding to get that drug, which is impossible. The other part of it is there's something called rare pediatric disease designation that Mr. Weber was referring to, and what that does is it's supposed to be an incentive for large pharmaceutical companies to create a drug for rare diseases. And the reason why is they can take this voucher, it's called, and say, for example, Novartis is coming out with a heart medication. Typically, it takes about 1.5 years for the FDA to approve that drug. Whereas if they get this voucher, then they're able to, after they cure a small rare disease like one of ours, then they're able to expedite that FDA process by up to 1 year. So if you think about a heart medication that can be worth billions of dollars a year, that's $1 billion they can make. So it was an incentive for them to do it. Unfortunately, all of our diseases usually fall under that radar, because if you're going to choose a disease, why would you choose one with 100 patients or 1,000 patients? You would choose one with 50,000 patients as an example, right? But the hope is that one day pharmaceutical companies will see the need and then move forward with these. Dr. Fink, do you have another perspective on that? I'm assuming.
I think, Terry, you said it. I have very little to add. I think you said it very completely. The comment is that why would a pharmaceutical company be interested in a very rare condition? And many times they're not. But I will point out that many people on this conference are probably taking cholesterol lowering medication, statins of some kind, or their spouses or their family members. Cholesterol lowering medication has revolutionized stroke risk and heart attack risk. So cholesterol management and the development of those drugs has been a worldwide transformation in the treatment of those conditions. And the discoveries that allowed those drugs to be developed were, and the investigators who found this won the Nobel Prize 30 years ago, or more maybe, but it was based on the analysis of very rare forms of hypercholesterolemia, very high cholesterol. And there are probably, I don't know, 50 or 100 families in the country that have that form of of cholesterol abnormality, very rare. But based on the analysis of those very rare families, they understood the process of cholesterol, of high cholesterol, and developed intervention to treat that, and that has been transformative. So the pharmaceutical companies may not really be interested in treating very rare conditions, except in the sense that they may serve as models to treat very common conditions. And so they have their eye on the very common conditions, and they're looking for a way to treat common conditions. And so the insight from the insight from analysis of very rare conditions may provide opportunities, or what they call treatment targets, for the more common conditions. What are these common conditions? Okay, spinal cord injury, stroke, degenerative nerve conditions, diabetic neuropathy, very common nerve degenerations. Insight into treatment of these very common conditions may come from the treatment, from the analysis of very rare conditions. And the hereditary spastic paraplegias provide, these are 80 or 90 different types of single gene disorders that cause a stereotypical nerve degeneration. And they provide each one is a wealth of information to understand what causes nerve degeneration. And that is what the pharmaceutical companies really want. And so it's that kind of long range perspective, I think, that it really attracts companies into this. And so we really have a lot to offer them. And because we really offer key insights into what causes nerve degeneration, how to stop it, how to prevent it, how to treat it.
Yeah, and I think if SPG4 or one of the more common ones was to come up with a treatment of some sort, I think it'd be a springboard for all the rest of them. I think that, you know, because I mean, Dr. Fink, how many people have SPG4? It's significant, isn't it? Well,
I mean, we estimated that there are probably 20, just based on on studies that were done in Europe, on the incidence of hereditary spastic paraplegia, and projecting those numbers into the United States population. It's estimated there are probably 20,000 people in the United States with a form of hereditary spastic paraplegia, probably more, but let's say 20,000. Yeah. And we would say probably 40% of that is SPG4. So that would be 8,000 people at least. I'm sure that anyways, at least approximately people with SPG4. But those are just estimates. But
yeah, because like I know I was talking with another family, and UMass has started an SPG4 program, and so is the Children's Hospital of Australia in Sydney, Australia. They're working on an SPG4 program as well through Dr. Leszek. SPG 15, you know, Karina Thurg has been trying for years to get that program going, and I think she's making some progress. And yeah, I think that there's so much potential, right? You know,
there's a lot.
I'm really curious about your thoughts around drug repurposing, Dr. Fink. Like, do you think?
Absolutely. That's critical because, you know, drugs, that's a very important way to go to find treatments. And for drug repurposing, there's two sources of it, or there are several sources of information, but one is a crowdsource. And this is a lot of data, and it's very difficult to sift through. And from crowdsourced data, you only get hypotheses that you can then take to the lab. You don't get you get ideas that to take to the lab. So for example, crowdsource would be, let's say everybody who's taking an antidepressant, and they say, I started this antidepressant, and within 3 weeks, I couldn't walk very well. As somebody, who, for example, who has hereditary spastic paraplegia or primary lateral, I'm making this up. This doesn't exist. This is a concept. Let's say everybody's taking drug X, and they say, I started this drug, I notice I'm worse. That's very important observation. And because that would say there's something about that drug, or let's turn it around. I started a certain antibiotic or a certain drug of some kind, and I started walking better. So we get this crowdsource information that would now say, let's do a clinical trial based on the information. So the one source of repurposing drugs is just by everybody providing information about their own personal experience, and we collect that information. That's one. Another source of information for repurposing is based on we have a type of HSP or PLS, and we know that this pathway is involved. Let's survey 20,000 drugs, 10,000 drugs that are known to interact with that pathway. And you can do this by computer. You can say we have a chemical library or a library of 40,000 drugs that includes 20,000 FDA-approved drugs. And we know the pathways that they interact with. And we have pathways of many, well, most all of the genetically known types of HSP. And we say, among these HSP biochemical pathways, which drugs have something to do with that? Either interfere with it or augment it or tangentially related or whatever. And then you develop a shorter list of drugs, 300, 400, and now one by one you test them in animal models. And so the animal models can be very simple at the beginning. They can be maybe not even animal models, they can be cellular models looking for a biochemical response. And then you take your best candidates from that and move them up to animal models and then into clinical testing. But long answer to your question, repurposed drugs, great, because you start out with drugs that interact with the biochemical pathway that have already been FDA approved.
Yep. Yeah, and I think that's one thing that I think that if we could, I think there's two important aspects of that. One is you focus only on the FDA approved drugs, because I mean, repurposing or making a new drug would take a long time. And the second thing is releasing the data of that information. I think that if we could do that, then we could take into computer models and then find better compounds.
I'll tell you one thing, interrupting you, I'm sorry.
Yeah, no worries.
That we already are doing this on a case-by-case basis. That is, every time there's a new drug that seems to have an impact on nerve degeneration or something like that, we are trying that. Unless it's very toxic or there's other restricting, and one of them was Ampera. Ampera was a drug that was approved for people with multiple sclerosis, and immediately it was tried in, as a kind of as a off-label indication in people with primary lateral sclerosis and hereditary spastic paraplegia, and other drugs too, other agents that have shown some promise in things like Friedreich's ataxia or ataxia in general, or Huntington's chorea, or even Alzheimer's. people are trying those drugs, or Parkinson's. People are trying other drugs in other fields to see if they would impact the symptoms in a kind of a off-label way. And this is common. This is not a study per se. It's not a clinical trial, but it's what clinicians do commonly.
So if we were to, here's an example, if we were to buy the Broad Institute Clue.io library that has all 7,500 FDA approved phase 1, 2, and 3 drugs, could we send, is that something your lab could run through? Or is that something that we'd have to get the Broad Institute to run for us? Not specifically for one disease. I'm talking about like, you know, maybe an SPG7 or SPG4 or something else, right?
Ask me that question again.
So basically, the Broad Institute, as of March last year, came up with a library called the Clue.io library. It costs about $15,000 to get, and it has every FDA-approved drug in phase 1, 2, and 3 and approval. So basically covers everything that you could repurpose, not remake. So the question would be, how do we get it to a lab that, because the biggest problem, as you know, is the assay. So just so everybody knows, when you find a drug, it's great that you have a drug, but you need to find a way to determine if it's going to work. So, you know, for example, if it worked, it would turn green. If it didn't turn, it would turn red. I'm oversimplifying, obviously, but that's the way you would do it in a computer model. So the problem is that finding these assays are hard to do, which some labs already have. So Dr. Fink, is there somewhere that we could start sending these drug screens to try working on those, you know, in the near future? I mean, for $100,000, you could probably run a screen somewhere, right?
Yes, we'd have to, in principle, you're right. And in principle, it would be very, very close in its direct approach, as you've described. We have to think about the logistics of it. And really, it begins with 2 points. One, as you said, and I'm glad we're having this discussion, and but it begins with what you said, you have to have a biochemical assay. And before, so in other words, what are you going to test? And before that, and somebody here mentioned SPG7. Let's talk about SPG7. Well, what are we going to test? SPG7 has several testable functions. And then it would be the same with any or similar with other HSP genes. What are we going to test and how are we going to test it? And are we confident that what we're testing is really the critical question?
Yeah, and the hope was, honestly, Dr. Fink, if we could get, for example, yourself and Dr. Blackstone, and you guys had essays for, say, 7, 4, and 15, I don't know, right? And we were to give you get this drug, maybe there'll be commonalities that are among, you know.
Okay, so we're just talking in theory, and
in theory, we're not talking like. You know,
what I would do, my answer to you at this moment in public, thinking about this, literally on my feet, is this.
I don't mean to put you on the spot.
No, that's fine. I enjoy being on the spot. Well, apparently I live on the spot. But what I would do is this. I would take one condition about which we know a lot. I wouldn't go 3 conditions. Now it's many things, and our certainty is much lower. I would pick one condition that we know the most about, and it could be even the rarest form of HSP. I don't care how rare it is. One condition that we know a lot about. And the one I'm thinking of, it could be SPG50. It could be SPG5. These are types of HSP that we know a lot about the biochemistry. SPG50, SPG5, for example, and there are others. Take one that we know a lot about and apply the screen to that one. Just one type, and see what candidates emerge.
So I'll save
I would agree with that.
I'll save you guys the money. SPG50 is already, AP4 is already doing it, so we're good there. So we should find a second one and get the.
No, I know. I remember the presentation about SPG50 and the candidate list.
Which is a great segue. Which is a great segue, if you don't mind. I've had somebody do a private message over here to me, and this person says, "I have a question.
Absolutely.
What is the significance of getting gene testing done with no treatment? What is the point of knowing which gene is mutated?"
Okay, very good question. And I'm going to answer this.
Okay.
for some people, not for everybody, but for some people, not having the laboratory confirmation is unsettling. It leaves a hanging question. Do I really have this condition? And yeah, I've been to this specialist and they gave me their opinion, and then I went to another specialist, they gave me their opinion, but I've got a life-changing medical condition that affects me and my family and my children, and I need to know more than someone's opinion. I would like to bring what we say closure to a diagnostic odyssey. Now, if a person feels they want closure, then getting a laboratory confirmation may bring closure to that question. It doesn't answer immediately. There's going to be another question like, now what do I do? And what does that mean? It doesn't stop there being all questions. It only answers that one question. And genetic testing is guaranteed to be expensive, and it's not guaranteed to always succeed in finding a gene, but it does often, not always, because sometimes it doesn't find it. Many times it doesn't find an answer, but if it does find it, one value is it brings closure to that question. The second question, the second issue is, does it help me get in line for clinical research, for a therapeutic trial? And I would say no, not really, because this is my opinion, because the day there exists a treatment for any type of HSP, a person could go out that next day and get gene testing done, and 3 to 6 weeks later, they would have that information. So I don't know that you need to get that information. It's like just-in-time delivery. You know, if it were available, if therapy were available, A, and B, if therapy was dependent on the type of HSP, then there's a two ifs. Then a person could go that day and get testing done, and very quickly they would learn the results, and they would know if it applied to them. And also, yeah. So I don't know that it would, because we don't have therapy today. I don't know that that getting a gene diagnosis translates today into a therapeutic advantage, but it does. The third issue is that it might expand the conversation. A person could say, oh, I have SPG 11, I have SPG 7, I have SPG 4. What kinds of things happen? Are there other people that are families I can talk to that have this, they're going through, that have children with the same type? I'd like to talk to them. In other words, there might be more information and network building and support group information based on that. But I don't think it would translate today into what we call actionable intelligence for clinical trial and clinical intervention.
I think to add to that, there's two more things that I think are important. One is, well, I'll tell you our story. When Michael was diagnosed, we did genetic testing, and we were told to go see the geneticist. And we were supposed to go, I think it was like November, but we didn't go until March. And those are 5 months that we'll never be able to get back, which we could have started our program. But I think the most important thing is, if you get tested and you get the results, at least you'll know what your outcome is going to be. Because you could have SPG 15 and just think it's going to be your body, but ends up being neurological or 7 or something else. Wouldn't you rather know that, you know, you have what your outcome is going to be, what that disease is going to affect? Is it going to affect your motor neurons, your arms, your legs only, your mind? I would want to know. And then on top of that, then you can go into natural history studies. People would understand the disease, and the more people that get enrolled into these natural history studies influences the companies to want to treat these diseases, right?
Which again is what we're trying to do, is to get folks to understand on our website. You're going to be able to find that information about genetic testing and what is available for you in that regard. And then we want you to be able to register with our foundation so that we know. Dr. Fink made a reference to a particular number, but we really don't have our hands around how many people in the world have this. When we did the August Awareness Week, we had this entire family of folks from Brazil that joined us. And then some of those were not even family members. So we had this entire population, you know, from these other countries that have gotten onto our annual conference discussion, our virtual conferences. If you don't mind, let me show that Brenda Lewis has been waiting patiently with her hand raised. I hope she's still on with us, and she may have a question. Miss Brenda? Miss Brenda, are you still with us?
Looks like she's muted.
Yeah. Well, Corey Wallet, we have your attention there. Why don't you go ahead and jump in at this point and talk with us about this?
Yeah, I mean, if there's questions, I'd almost rather ask questions, but I love the conversation between Terry and John. I mean, this is the heart of the issue, right? It's not easy, it's not fast, and we're trying to do it fast and easy, but I really like the sentiment of, you know, all of this takes money. And I didn't really appreciate before joining the board, I don't know, over a decade ago, how important that was and how difficult that is and how important that really is for all these families. So, you know, to me, I just want to do the best job that I can, helping to communicate science that's complex and sometimes hard to understand in a way that we can all understand, because that's definitely possible. That's what I like to do. And if there's questions, happy to help.
Yeah, Miss Brenda didn't jump back in, so I don't know if she has left the phone or left the screen for a few minutes, but that's why it's so important, you guys, for us all to work together. Even though $5 here and $10 there, you know, seems like, you know, a very little amount of money, together collectively, it really does, you know, have some benefit for us. I think, Miss Brenda, have you joined us?
Yes.
Hi. So, you had your hand raised, and thank you for waiting.
Well, I was just having my hand raised to say hi to everybody, but I do have a question for Dr. Fink. He loves me because I always have a question for him.
Go right ahead, please.
So from your professional doctor standpoint, what do you think is going to be the cure for this in the long run? Do you think gene editing will do it?
I would hope, it would be my hope that it'll be small molecule treatments. That would be my hope. That would be, I think that initially we're going to go through a phase of gene therapies. And I'm totally in support of this, and we're developing an animal now in which we want to try some gene therapy for. But but I think we're going to go through a phase of gene therapies. But I think my hope would be that we would end up with small molecule interventions. And for example, how do we treat Parkinson's disease? Right now, we're not using gene therapy for Parkinson's. I mean, except in some experimental situations, we're using small molecule replacement of a chemical that's deficient. And I would like to see us move in that direction. But that kind of small molecule intervention is moving at a much slower pace than gene therapy trials. And so we need to get to success and then build on success. And I think that gene therapy promises the greatest success. And as Corey said, it's not fast, it's not easy, and it's not inexpensive, but it's faster than other approaches. And I think we may get to first base that way. And then in those gene therapy trials, we learn, as it was mentioned by Mark Weber, that an important part of these trials is having the clinical outcome parameters. So when we do gene therapy trials, well, by necessity, we have to have had, at least for those patients, outcome parameters defined. So not only do we test a particular type of HSP, and not only do we test a general approach to treating HSPs, for example, gene therapy, but we advance the process of clinical trials in HSP per se. And so the short answer, and I've never been accused of giving a short answer, But the short answer would be, I think that I think we're going to go in phases. I don't think that one approach is to the exclusion of another approach. I don't think one size is going to fit all. I think different types are going to have different treatments. And I think initially the near horizon therapies will be gene therapies, and the farther horizon therapies I think will be chemical interventions. But that's just how it seems to me at the moment.
Is this going to happen in my lifetime?
I can say, God willing.
God, I hope so. Because I was diagnosed 14 years ago, and they told me 10 years ago they'd have something soon. And it's frustrating, and it's frustrating for everybody.
These things never happen the thing about it is, and I've known Mark Weber a long time, and I used to say 5 years. Well, I stopped saying that because 5 years has come and gone several times. So I don't put a time on it, but I'll put it like this. So you can't really make a prediction because science gets to a tipping point and then things happen very quickly, often. And so we don't know if we're at a tipping point or getting to a tipping point. And so I don't know. And it's not just the treatment of these conditions. It's what's happening in treating Alzheimer's, what's happening in treating diabetic neuropathy, what's happening in treating stroke or spinal cord injury. In other words, all of the field of neuroscience and neurodegeneration and nerve injury, all of these fields are growing. And trust me, as soon as there's an advance in one field, we're going to capitalize on it and apply it here. So all of the fields are growing. And all these drug discovery and repurposing, this is happening in neuroscience broadly, especially for conditions like Alzheimer's. There's a huge market for that. And a lot of drugs are being tested and considered for that.
And remember, the one thing about gene therapy is there's a lot, like, listen, gene therapy is it's a poor treatment, right? I mean, you're affecting, if you're lucky, 5% of the brain. Then you have to worry about immunoresponse, which then is, you're most likely going to be on a immunosuppressant the rest of your life, which may interact with the drug that you're probably taking. There's a lot of problems with gene therapy, right?
Well, has CRISPR been interested in HSP at all?
The problem with CRISPR is, if you were to, first of all, as an adult, CRISPR would probably never get approved unless you have a life-threatening illness. And the second thing is, CRISPR in itself has too many, they call it offshoots. So you might go to cure HSP and cause a different disease. There's some other amazing things like prime editing, which is done by a company called Beam Therapeutics. They're doing some amazing things, but if I was to estimate, I would say that any kind of gene editing in a large scale, baby comes out, person gets diagnosed, whatever it may be, to gene editing is probably 7 to 10 years away, if that.
Doing a lot of work right now. I work for a company, and my lab, we run over 50% of the clinical trials in the world. That's what my company does. And I just do the genetics part. So I just do a little tiny part, and we're doing a lot with CRISPR. And I think what you're saying, Terry, is right. But where most of CRISPR is used right now is, for instance, with eye diseases, where you can actually have access, and it's miraculous when that happens. Same with some gene therapies, but where there are some rare blood disorders you may have seen in the news. Sickle cell has been worked on with CRISPR, because you can take bone marrow out, you can treat the cells, and you can put bone marrow back in, right? That's something that we do medically and have for a long time. It doesn't mean it's easy, doesn't mean it's not painful, but you can actually remove that tissue, treat it, and put it back in. And with neurons, that's not possible at this point. But what is being done with CRISPR, like everyone has said, we're going to learn quite a bit from it and be able to apply it in new ways in the future. So it will be a future direction. Doesn't mean that it's a direction that's being pursued as aggressively now, mostly because of the targeting and tissue pieces. But it's really important to make those learnings now so that we can know where we can apply it in the future.
And gene therapy has two parts to it. It's the putting the gene in or editing the gene, and then delivery. And the delivery piece is the hard part, is delivering this gene edit or gene modification, whatever it may be, to where you want to. Our gene therapy is using AAV9, but that's, again, it's a poor delivery mechanism. And until they do, like, for example, nanoparticles or some other delivery mechanism to get it to more of the body or more of the brain, then it's a limited piece, but it's the best thing we have right now. And I wish there was a drug, because you pop the pill in, something goes wrong, you stop the pill, right?
I want to just interrupt for a second, because I've got to run to a meeting. I'm overdue for another conference, so I'm sorry I have to leave. But I just want to say, we have 47, 48 people listening, rapt attention to discussions about therapy, because we need treatment.
Yes.
Right.
That's what this whole thing is about. We need the research to develop the treatment. And that's what this Giving Tuesday is all about, is to take us from these conversations that we are riveted by into conversations about, well, now I'm trying this agent. What do you think about that? We don't want to have this conversation about what the future of trials, when will the trials be? We want to have the discussion about trials, plural, that, that we want to move this to the next level. And that's what the Giving Tuesday is all about. And so I wish everybody the best of luck with that, but I'm sorry I have to go. But thank you very much.
Thank you,
Thanks,
Dr. Fink.
Thanks, Dr. Fink.
That is a great segue into our ending of our first Giving Tuesday Zoom. I couldn't have said it any better. I really appreciated Dr. Fink taking that moment there. But I'd like, before we get off, I'd like to let you know the other folks that are on the call that are also on the board of directors. You heard from Frank, you heard from Jim, you've heard from Mark, you've heard from Corey. I'd like for John Cobb to say hello.
Yeah, and actually, thanks, Norma. I'll take this moment to ask a quick question, which is, let's say I tomorrow do want to do a fundraiser. I know Classy has been discussed, and I know Facebook has been discussed. And obviously, the more time we put into it, the more money we're going to get out of it. But if I'm tight on time and I want to whip something together real quick, how long would it take me to set up a fundraiser, and what would the easier / better platform to do that be?
If you want the help of the foundation, then the best thing to do is to send us maybe a paragraph or two about what you want to do, how you want to do it, what is the goal dollar amount of your fundraiser. Send us a couple of pictures, or we can pull some stock photos, from wherever. And then before probably 2 hours, we will have that online for you, ready for you to share that link so that we can start raising money.
And that would be through Classy?
Probably so. That would be one of the easiest platforms. But if you would let us kind of help host that for you, then it will go straight into the bank account of the foundation. And then it doesn't have to go through you. Then you send it to us. It's just an automatic deposit in.
Gotcha. And then that would be promoted by creating a Facebook post and sharing that?
Yes. And we could even, if it's actually like a timed thing that you wanted to try to draw in a bunch of folks, we at the foundation, we run several Facebook pages, Instagram, Twitter, we have all those platforms, and we're looking at expanding those as well. But we could actually create for you that event page on our Facebook page, make you the host, so that you can also gather and answer any questions that might pop up. But then you can share that to your family and friends and be able to have that information there readily available. So then all you got to do is add those links to the Classy platform inside the Facebook link.
Okay. And if I want to customize it at all, do I have the ability to do that?
Well, Facebook you will. The Classy platform, you will have to let us help you do that.
Okay. Awesome. Thank you very much.
Yeah, it's real simple. So just send us an email with photos, whatever the blurb is, just a couple of paragraphs. Give us some information to work with. Then we'll create some flyers and graphics to go along with that. If you have the ability to create your own graphics, just send that to us, and then we can even make it faster for you.
Awesome. Thank you.
Yeah. Yeah. So as you guys saw tonight, it's very important that we get you to register so we know who you are. If you've not already done that, you then will begin to receive the emails from our foundation, from all the people that are working on that. You'll begin to see e-newsletters. You'll begin to see some updates of information. But more importantly, your information will be on, you know, our database, so that if the doctors do come up and they need a particular SPG, then you're going to be able to have your name on the list. I'd also like to take a minute and introduce Tina Krogan, and her better half, or other half, Tim, who works with her very closely. Tina, please say a word. Tim, y'all say hello.
Hi there. I'm Tina Krogan. I'm here in St. Louis, Missouri. And Mary Schultz is also here. She is the ambassador in the St. Louis area.
And I think we had, thank you so much, Tina. I think we had Karina on from the UK, but she had to go. She works with her daughter, the foundation, the Maddie Foundation. So she had to go. I don't think I've missed Dina. I think Dina Landfair, she's on. She's chair of our education committee now. Tim is the marketing chairperson. So, you know, we have a couple of 3 or 4 committees that we have kind of created so that we can help bring the work of the foundation, you know, right into you, and then you work with us. So if you have an interest in fundraising, if you have an interest in marketing, if you have an interest in education, you know, like training, if you'd like to be an ambassador for your community, for other folks that are maybe near you, you know, we're not really per se counselors, but we're support. And that way we can talk to other folks that may be in our communities or in our state that maybe they're just newly diagnosed and they need a friend. So that's what I count all you guys. I count all of you friend and family. So if you guys liked what we've done tonight, then we have appreciated you more than you absolutely know that you took time to join us. And I would like for us to periodically try to have some more of these. Not that we're talking about Giving Tuesday, but maybe we're going to be talking about something else. But tonight really was about Giving Tuesday, and Dr. Fink, he really put it right on it. And that is that, you know, together we've got to find ways to raise the awareness and raise the funds so that we are at the table for the medical research. So unless there's nothing else to say, which I could talk all night, and I appreciate your listening to all of us, we're going to call it a night. We're going to thank you for your time and energy, and please donate to Giving Tuesday, noting the Spastic Paraplegia Foundation tomorrow or tonight before you go to bed.
Thank you, everyone.
Thank you.
Thanks, guys.
Thank you.
Thank you, everybody.
Thank
Good night,
you.
everybody.
Thank
Thanks.
you.
Good to see you all.
Take care.
Take care.
Bye.
Good night.
Bye.
Good night,
Thank
everybody.
you.
God bless.
Thank you. Good night.
Be safe. Let us know if we can help.